Well, hello, AP Bio students. Mrs. Shelton here, and we are going to be talking about unit number 4. So this is our AP Bio review for unit 4. So this is cell communication and cell cycle. Now, with this particular unit, it's actually parts of our textbook chapter 5. So I think it was chapter 5, section 6 was the cell communication, and then cell cycle was chapter 9, which was cell cycle and mitosis. So we're just going to address the topics that College Board wants us to address for unit number 4, but this is back in the test prep series. Topic 2 is where the information would also be a good review for you in addition to that 1.1 chapter. So for cell communication and cell cycle, there's lots of illustrative examples of cell communication that we're going to just address. But again, with illustrative examples, you need to know some examples. You don't need to know all of them. So don't freak out if what I show you are things that we haven't talked about or you don't understand. If you understand parts of them, perfect. That's all you guys need to make sure that you are addressing. Now, in this particular unit, what they're talking to you and what they want you to develop an understanding for is just the role of cells and focusing on how cells use energy and information transmission to communicate and to replicate. And then take that on to Unit 5. Now, some of the big idea questions that you should be able to answer after understanding this unit is big idea 2 on energetics, as far as understanding how cells use energy to communicate with one another, and also understanding this information storage and transmission. So understanding the cell cycle and also then ways that cells communicate with one another as well. Now, for preparing for the exam, some of the tips that College Board does share with us as far as using this unit to prepare for the exam. With this, it's understanding the significance of the steps in cell signaling. And it really is through this transduction process, the amplification of the signal. So all it takes is just a little bit of a signal molecule to then get a pretty big response inside the cell. So understanding then how this works with drug interactions along those lines, or any chronic disease arises when there is this problem with cell communication. Now, also too in this unit with cell cycle, it's making sure that we understand our cell cycle checkpoints and control of the cell cycle in order to be able to talk about that. And then also being able to compare mitosis and meiosis are also important as well. Now, our overview of this unit, cell communication, cell signal transduction, again, signal transduction, transduction pathways. We're going to look at some illustrative examples. We're going to understand the basics here, but it really is not anything too complex that we need to worry about, as long as you're understanding the basics of why do cells need to communicate, and you understand the 3 basic steps of signal reception, transduction, and then response. We'll talk about feedback, negative and positive feedback. We'll talk about the cell cycle, so making sure we understand our G1, our S, G2, and our M phase. And then we'll also talk about that regulation of the cell cycle as well. So when it comes to first topic 4.1 on cell communication. So the ways that cells can communicate with each other is through chemical signaling, long distance, short distance, cell-to-cell contact. But with this, and here's just a diagram that kind of just helps you remember, here's contact-dependent cell-to-cell communication. We have to have these receptor proteins that are on the membranes of the plasma membrane of cells. And so it's through these that cells can communicate with each other. Now, we can have through long-distance endocrine responses where hormones are sent through the bloodstream in order to now reach a target cell. Target cells have the correct receptor to match the signal molecule. We can have paracrine signaling, which is a close contact local release of a signaling cell. This would be an example such as like an inflammatory response. So where local histamines are released in order to now start increasing the permeability of surrounding capillaries. We also have neuronal responses here. This is how 2 neurons communicate with each other through, at the synapse, releasing neurotransmitters that now the dendrites of the next neuron has to have the correct receptor in order to propagate that signal. So those are just some examples. Now, some other illustrative examples. Here's one where I do get a little sad because we didn't get a chance to talk about the immune system, and the immune system is freaking awesome. And we didn't get a chance because we left for home isolation. We didn't get a chance to talk about our MHCs, class 1 versus class 2 MHCs, and how this is how your body recognizes self versus non-self cells. It again is pretty freaking awesome. But what ends up happening in our immune system when we have the specific responses in our acquired immune response, what ends up happening is we have cells that will present antigens out the class 1 or class 2 MHCs. And when they are antigen presenting, so they take a piece of antigen fragment and they shove it out through these MHCs, these membrane proteins, well, that changes it's kind of like a barcode, and it ends up changing what the protein combination is on your cell membranes. And that's what your immune cells go, you don't belong. So, this is actually how helper T cells and cytotoxic T cells will have this direct cell to cell communication with your own infected cells. And so, it knows if your cell is infected with a virus. You have cytotoxic T cells that know, oh, yeah, sorry, gotta kill you, because it's antigen presenting and they have this cell to cell communication. Helper T cells also get the signal in order to actually begin the immune response through these antigen presenting macrophages. So these dendritic cells that are now presenting. So that's just an example of a cell to cell communication that's in our immune system, helper T cells and killer T cells, in order to make that direct contact. Another example that you have had is with plasmodesmata. When we talked about cells in our plant cells, through the cell wall, there's these pores, these plasmodesmata that allow materials to move from one cell to the next in order to have a cell to cell communication. Now, some other cell communication using local regulators, neurotransmitters from neuron to neuron at the synapse. So, where this would be the axon terminal, and at the axon, neurotransmitters get released, and the dendrite has to have receptors. And these are actually ligand gated ion channels. So, that's just carryover from a previous unit. And so, when enough neurotransmitters signal and open up these channels, well, sodium ion channels rush through. That changes then the voltage, which now propagates the action potential down the rest of the neuron. Plant immune responses. We also ran out of time and we didn't get a chance to talk about plants and their immune response that happens. But they have receptor proteins that can actually recognize pathogens. And so this is an example of this cell to cell communication where, with this recognition, it now actually triggers an immune response in order to now ward off infection from other cells. We also didn't talk about quorum sensing in bacteria yet. And really, what this is referring to, we did talk about the fact that bacteria has the ability to uptake DNA. They can also form biofilms as far as grouping together. And so it's actually when bacteria become very, very high density, they actually have this ability to communicate together, and so they can send signals to each other when they have this high cell density, which is also referred to as a quorum, or this quorum sensing. So they end up having this group behavior of all of them doing the exact same thing. Morphogens in embryonic development. When we did talk about embryonic development, and we talked about the fact of what are some of those outside triggers to initiate transcription, translation responses for cell differentiation. Well, a lot of it is concentration of the cells that are existing. So morphogens would be these chemical concentrations. We talked about cellular determinants. And it's some of those that are going to, based on just the environment of the cells, and now these concentrations of these morphogens are going to trigger different cellular responses. Some other specific examples, here are some hormones just to be familiar with. I gave you a picture here of your pituitary gland that produces some of these hormones, but obviously human growth hormone is going to have a different response because it's for growth and elongation of bones and development of more connective tissue, versus then thyroid hormones. Your thyroid hormones are going to be controlling your metabolism, where now testosterone or estrogens, well, those are going to be controlling then your secondary sex characteristics. So it's recognizing these long distance endocrine hormones do have different functions. Insulin, you're all very familiar with, produced in the pancreas. And so, one of the actions that insulin has at the cellular level, insulin travels through your bloodstream. Well, in cells, then, the insulin receptor is triggered by the insulin, which now allows for this transport of glucose into the cells. So, here's just again an issue if this receptor doesn't work anymore. So there are receptors that no longer work, and so then it doesn't respond to the insulin, or if there's not enough insulin produced, then also you don't have the cells responding. So when we do talk about our introduction to signal transduction, so with signal transduction, it's understanding with this, we have to have obviously reception, and then we have our transduction, and then we have our response. But with this transduction, it's understanding some of these terms here of, again, what a ligand is. So a ligand is a molecule that is a signal molecule that has a specific target, and the target has the matching receptor on it. But understanding a G protein coupled receptor, some of these signaling cascades that happen too. So when we're addressing these secondary messengers or ligands, they're just different ways that reception and then the initiation of a transduction can occur. Our three basic steps of signal transduction pathways: we have to receive the cell has to receive the signal. There's a receptor protein that matches the ligand, the hormone, or the environmental stimulus. Then we have a biochemical pathway of transduction, and the whole purpose of transduction is to amplify the signal, and then the cell is going to have some type of cellular response. Cellular responses involve either the initiation of transcription or it's going to be a change in a protein in some way, shape, or form is what's going to happen. Here is a transduction pathway. So here I have reception, but here's my transduction. And oftentimes we have like a phosphorylation cascade is what it gets referred to. And in phosphorylation, we're phosphorylating, which means we're transferring phosphates onto these active enzymes. And so when I have a kinase, remember kinases are enzymes. Anything that ends in ASE is the name of an enzyme. Kinases are enzymes that phosphorylate. And so here's just this chain reaction of phosphorylation in order to activate a particular protein is the end response. Now here would be a G-coupled reaction and a G-coupled reaction. So here's my reception. But what this is doing is it's converting GDP to GTP, which now is actually triggering this in order to now create a secondary messenger. So now I complete my transduction. And so it's just recognizing there's different ways to receive. There's always, if this is a polar molecule or then reception is on the outside. Do remember too that I can have internal receptors, and my internal receptors will respond to steroid hormones. A steroid hormone then is nonpolar, and being nonpolar, it can actually just diffuse right through the lipid bilayer, and then it can meet the receptor that's inside the cell. So keep that in mind and why they labeled this a non-steroid hormone. That's more important than understanding G-coupled or secondary messengers. It's all part of starting that transduction in order to get to the response. Now, more information than you ever need to know on signal transduction. So with this, it's going to result in changes in gene expression and cell function. We already talked about that, but understand just some different types of signals. There's some environmental factors that can be the actual signal that now gets received. So in plants, the greening response that actually happens in plants, well, it starts with light. So there are these receptors, these phytochromes that are activated by light that can actually penetrate right through the lipid bilayer. And that is going to now trigger my transduction that is now going to initiate a response. And in this case, you're seeing a typical response, which is to initiate transcription. And once I initiate transcription, then I get translation, and now I get proteins that affect it. Now here is also another action here. So here I have an ion channel, and this ion channel, if it's open, this open ion channel now is going to trigger a transduction pathway that leads to transcription, yada, yada. So recognize some different types of signals that could, some environmental signals that actually can initiate a transduction and then lead to that response, which is a change in a protein in some way, shape, or form. Now, again, College Board does not expect you to memorize any type of specific reception, any specific transduction, any specific response. What you do need to know, though, is always signal transduction pathways always include reception, transduction, response. If they're giving you anything, they're going to explain it to you, and read the background information they give to you is really what you should be doing, because the background information they give to you is going to give you the details that you need to know, that you need to be able to analyze and assess any changes that they then describe for you. Now, some illustrative examples using signal transduction to respond to the environment. I already talked about quorum sensing, so the high densities of bacteria where all the bacteria can now respond and act the same would be your quorum sensing. But here's just another example here of epinephrine. So with epinephrine and this fight or flight response that we actually have, and we release epinephrine from the little adrenal glands on the tops of our kidneys. I usually refer to them as the little party hats on top of our kidneys. So when we have that and epinephrine is released, well, what's going to happen is it has different actions on different cells. Some cells are going to receive epinephrine, and what that is going to do is it's going to trigger then the breakdown. So once it's received, because it's a different transduction pathway, like in your muscle cells versus now in your intestines, what's going to happen is my muscle cells are going to start, or my liver cells, they're going to start breaking down um, glucagon or glycogen into glucose in order to increase the amount of sugar and amount of cellular respiration that happens, because my muscle cells are going to become highly activated. But a different response then is in my digestive system. Cells in my digestive system are actually going to shut down, and they're actually going to stop using energy, is what's going to happen. And how I can get different responses from different cells is, even if it's the exact same signal molecule, same receptor, the different response comes from different transduction pathways. Here is cytokines. This is, again, since we didn't talk about the immune system, we didn't talk about cytokines. Cytokines are these chemical signals, and these chemical signals are actually highly involved. So these signal molecules in our immune system responding, and also cell generation and cell response, wound healing, etc., are some of those biological effects that cytokines have for cell replication and division as part of an immune response. Another illustrative example, yeast cells. We also did not talk about mating yeast cells. And so with this, it's just showing these chemical signals. They have to receive these chemical signals. So here from the female, from the male. And so this now goes, oh, hello. And so once they both receive the signal of mating, then they can actually have this cell-to-cell contact, the agglutination, and the successful mating. But it's all, again, through signaling and through that reception, transduction, and response. We did talk about the SRY gene on the Y chromosome. So, but that Y chromosome is producing proteins. Those proteins are then signal molecules that then these tissues in your cells or in these organs actually receive the protein signals. And so that is the difference between the development of testicles versus the default, which is the development of the female state. Ethylene levels in fruit ripening. Now, this is going to be an example of a positive feedback as well, but it is actually the reception of ethylene, this gas, that leads to signal transduction, leads to a response of now ripening and increasing the amount of sugars that are in plants, but it also then triggers the production of more ethylene, that now that gas travels to other surrounding cells and that increases the ripening of all of the fruit at one time instead of just certain fruits and some fruits on one particular plant. Because if you are trying to bring lots and lots of animals to eat your fruit so you can spread your seeds, well, we want all the fruits to be ripe at one time. And so this ethylene production that ripens the fruit, but then it also becomes a positive feedback because more ethylene and more ripening leads to even more ethylene. Now, lastly, we talked about Hox genes. Hox genes, remember these particular genes that are for determining body spatial arrangements. So that would be another illustrative example, just to understand, just again, more just cell-to-cell communication and why cell-to-cell communication is important. When you have trillions of cells, they have to communicate with each other in a myriad of different ways. But you got to receive a signal. There's a transduction pathway that amplifies that signal, and then there's a response. But you know this. Yep, here it is. Changes in the signal molecules can affect the signal pathway. Do we need to talk about more than that? If my signal changes, then I don't have the same thing. If my receptor protein changes, then it can't receive the signal. If there's something that changes in the transduction pathway, I don't get the same response. So remember that when we get changes in any of those steps, I'm going to get different responses, and just be aware of that. But this is also where drugs interfere. So whether it is a pharmaceutical that is trying to stimulate a particular transduction pathway and response, or whether it's there to block a particular reception and transduction, kind of understand that basics here. So again, I've got my reception, I've got my signal transduction, and then I've got my response that's going to be happening here. And any changes in the receptor, in the signal, are going to change the response. Not that tough. Okay, so feedback, positive versus negative feedback. This is how we maintain homeostasis with negative feedback mechanisms, and then why we need positive feedback systems. When we do talk about our negative feedback, what is happening is we're always trying to maintain a set point. We're trying to maintain homeostasis. So our positive feedback, we're always trying to increase that stimuli in order to get a change in that situation. So examples of positive feedback, childbirth, blood clotting, and fruit ripening would be examples of positive feedback loops. Regulation of body temperature, of blood pressure, of amino acid levels, of hormone levels. Those are all negative feedback loops in order to maintain homeostasis. We also talked about controlling biochemical pathways with feedback inhibition. And in biochemical pathways, oftentimes the end product becomes an inhibitor of one of the initial enzymes. And so now as concentration increases, that shuts down the enzyme that now stops the biochemical pathway. But now as that product concentration gets lower, well, now that product is going to move from and off this enzyme 1 back into solution, which now activates that enzyme. And so now that's going to trigger the biochemical pathway again. But that's my feedback inhibition at the biochemical pathway level. Some illustrative examples. You guys know the role of insulin and glucagon in maintaining the set point of blood sugar. Insulin, ultimately what insulin is going to be doing, if you went to Dutch and you decided that you wanted your 100 grams of sugar wrapped in a fruit icy thing, also known as a rebel, you're going to have high amounts of glucose in your blood. So you need to now produce a lot of insulin, which now tells your liver to bind those monosaccharides into glycogen. Now, what's going to now happen is you should, if it was an ideal situation, you should be back at your set point. Oftentimes, however, because of a high increase in glucose, way too much insulin is then secreted, way too much of the sugars bind into glycogen, your blood sugar drops to a lower level because you've bound up too many glucose molecules. So now what you have to do is wait for glucagon, and glucagon is a hormone that's now going to trigger glycogen to now be broken back down through hydrolysis reactions back to simple glucose molecules to achieve a set point. Anytime we're talking negative feedback, there's always a system for bringing things down to the set point and a system for bringing things back up to the set point. When we talk positive feedback, some examples of positive feedback would be lactation in mammals. This is how you can start with a baby at 6 pounds and still be able to feed a baby at, when they're 30 pounds, or a mother who has twins is able to still produce enough breast milk because of that positive feedback. The more milk that is being used, the more suckling that ends up happening, the more milk production is going to occur. Ripening of fruit, ethylene. When ethylene is produced, that triggers this positive feedback response of more ethylene, more ethylene, more ethylene, and we get all sorts of amplification of the stimulus. Childbirth is another one. That release of oxytocin to initiate contractions. That stimulates more oxytocin to stimulate more contractions. And we also talked about blood clotting, that clotting response in order to amplify the clotting process until you stop bleeding. Now, lastly, cell cycle. You guys should feel very, very comfortable with the cell cycle. It's pretty easy and straightforward, making sure we're understanding with the cell cycle, our cell cycle. All cells just continuously cycle through this unless they go through a G sub 0 state of a non-dividing state. So when a cell actually is first produced through the M phase, mitosis cytokinesis, that cell is going to go through an initial growth phase, and increasing its cell contents, increasing its size, but it's still doing its normal thing. And its normal phase is called interphase. And so G1, S, G2 are all part of interphase, but this is also at G1 where a cell could enter the G sub 0 phase, but also it cannot pass if it doesn't meet this checkpoint. There's a checkpoint here at G1 that we'll talk about. Then remember, the cell is going to undergo the S phase. It's the S phase where DNA replication takes place. This is a very, very long phase. Very important that you remember that DNA replication happens way back here. It doesn't just magically all of a sudden occur right at mitosis. Mitosis is just division of the nucleus. So remember, at this point, my DNA content from here to here, my DNA content doubles from G1 to G2 during the S phase. And then it's going to go back down to normal amounts during mitosis. So it's going to be normal amounts of DNA here at G1. It increases during S, and then my DNA content is going to be constant here, but double the size. And then cell division is going to take place during mitosis, and that's where my DNA content gets produced. So remember that my S phase is all about my DNA replication. My G2 phase is going to be a secondary growth. I'm going to double check that all of the DNA is there. We're also going to have a little extra cytoplasm, a little extra organelles, because when the cell divides, we're going to divide the nucleus through mitosis, and then we're going to divide the rest of the cytoplasm, all the organelles and cell contents, through cytokinesis. Remember, with mitosis, I've got PMAT. Prophase, metaphase, anaphase, telophase are my 4 steps. It is very coordinated. Mitosis is the division of a nucleus from 1 diploid nuclei to 2 diploid nuclei, or 1 haploid nuclei to 2 haploid nuclei. In mitosis, it is it's nuclear division resulting in 2 identical nuclei, and it's identical to the original parent cell. Now remember to count chromosomes correctly. In this picture here, I have 4 chromosomes. In this picture over here, I have 4 chromosomes. But remember that my chromatid, so here I've got 2 chromatids that are attached at the centromere. And so this 1 chromosome is actually double the DNA content with 2 identical sister chromatids attached to the centromere. So remember, metaphase, my chromosomes line up on my spindle fibers on my metaphase plate. Anaphase, my sister chromatids now detach. And now at telophase, I get 2 new nuclear envelopes forming around my chromosomes. And then regulation of the cell cycle. And the last thing we need to talk about for this particular unit, when we regulate the cell cycle, remember our checkpoints and the role of cyclins and CDKs. Now, it tells you right here, you do not need to know the specific cyclins or CDK pairs. However, you should in general understand why we have checkpoints at different parts of the cell cycle, what happens if there is a disruption in those checkpoints of the cell cycle, and then just in general, a cyclin and a CDK and how they work. Now, in this particular diagram, I am showing you lots of different cyclins and CDKs, but let's start with our first checkpoint. So here's where my first cell would be created, but here's a checkpoint at G1. At G1, I have a checkpoint, and a cell is not going to move onto the S phase where it replicates DNA unless it passes this checkpoint. Well, this cell, remember, had to be doing its normal cell job. And so during its normal cell job, it also needs to be producing a molecule called cyclin. And so if there is enough cyclin produced in the cell, it is that high concentration of cyclin that can now trigger this enzyme, this cyclin-dependent kinase, to now trigger the actions that can move the cell into this S phase of replication. Again, G2, before mitosis happens, we want to check to make sure that the cell is ready to divide and is ready with all the right chromosomes, with the amount of energy in order to be able to divide. And then there's another checkpoint at M, at this M phase, in order to make sure that after the M phase, it's ready to undergo cytokinesis. So these are my checkpoints. And at every checkpoint, it's my cyclin concentration. My cyclin concentration is high enough, then that triggers a cyclin-dependent kinase to now start a biochemical pathway that can move the cell into the next phase. So remember I told you guys in the analogy that I gave you, it's like the water in the bucket at Sunsplash. The water is the cyclin, and the water builds up, and the water builds up, and the water builds up, and ultimately when the concentration of water is high enough, that triggers the bucket to now act and to move over. So remember, my CDKs are constantly there in the cell. It's my cyclin levels that are going to be lower or higher at these checkpoints. So cyclin-dependent kinases. Now, when something goes wrong with the cell cycle, well, some things can happen. So remember with cell cycle controls, some cells, they have anchorage dependency on what medium or extracellular matrix that they are growing on. There's density dependency. So when cells are now communicating and butted up with other cells, that stops them from growing. But when we have cells that are no longer abiding by cell cycle regulations, we now get either cancer cells, or we also get sometimes cells that just won't even produce normal cells. And instead of having a normal programmed cell death, because this does happen in our cells, we have programmed cell death, apoptosis, but sometimes there's enough damage in the cell that it just, oh, we made a new cell. But cancer is usually the result of cells that are no longer abiding by normal cell cycle regulations, is what we need to make sure that we are taking out of this. So for Unit 3, signal transduction pathways, if you got down reception, transduction, response, you're good. You can apply that to anything as far as how cells communicate, either long distance signaling or short distance signaling. It's all the same. We have to have the reception of the signal. We have to have the transduction pathway that amplifies the signal. And then we have the response that is different. And then also remember our cell cycle, and remember then our mitosis and our phases of mitosis. So with that, hopefully you guys understand unit number 4. You're feeling comfortable with unit number 4, and I will talk to you guys another time. So thanks for listening. Bye.